Our second video is going to be focusing on the first part of photosynthesis, which is light reactions. You may also hear it called light dependent, which means it needs light in order to operate or run. Let's just jump right into this. So this diagram has a lot going on. The first I want to point out is the equation at the top. So you do need to know the equation. So you need to know the reactants and what does it produce. And there's different ways that you can write this, and I do want to point out a couple. You'll always see the reactant carbon dioxide and water, 6 molecules of each. And then it might go just the arrow and then show the product. So they might write light and energy above and below the arrow, or they can include it as the reactants. And then the products is going to be C6H12O6, which is glucose. And we know this is a sugar because going back to our chemistry unit, it has the ratio of 1:1, 1 carbon, 2 parts hydrogen, 1 part oxygen, which makes it a sugar, C6H12O6. And the other product it's going to make is 6 molecules of oxygen released to the atmosphere, which animals, which include us, we will need. So all these other diagrams, we kind of pointed out in the other lecture with the structure that you've got your leaf, and then here's the cell, and then we're going to focus on the chloroplast. So this is inside the chloroplast right here. And these are, if you remember, these are the thylakoids. So these are the thylakoid membranes. Inside the thylakoid membranes is where light reactions will take place. And then the stroma is around it. And then we're going to also look at the dark reactions, and that is called the Calvin cycle. That'll be our last lecture. So you may also remember from the previous lecture that this is a thylakoid membrane, and inside the thylakoid membrane is where light reactions are going to take place. So photosystem consists of two pathways, or two parts. So we have the light and the dark reactions. Here's our equation again. Do make sure you know that. This is another way to write out the equation, actually, instead of writing out carbon dioxide light energy. So this case, we're going to include water because water is going to be cycled throughout this process for both light and dark reactions. So we have carbon dioxide, and that is what animals, including ourselves, breathes out. So the plants are going to use this. So it's a symbiotic relationship between plants and animals. So animals need oxygen, and that's what plants give off, and plants need carbon dioxide, and that's what animals produces. So they've got this continuous cycle. And I kind of touched on the guard cells and the stomata, the opening and closing, allowing those gases to go in and out. So again, make sure you know what these images look like, and you can identify if it's open or closed. So what really happens is through osmosis, we have water either coming in or going out of these guard cells on each side of this opening or pore. It seems a little backwards, but when water comes into the guard cells, okay, it's going to make them really rigid. And when it makes them rigid, if you hold your hands really tight, okay, it's going to make it open. And when it relaxes, okay, it softens up, water goes out, then it's going to loosen up, and then it's going to close. So if it's tight, okay, it's going to open. If it relaxes, it's going to close. Is how you can remember that. So water moving in or out. So we can tell that this particular microscopic image, that this one is closed. There's like a slit, that little cat eye I talked about. Okay, so that would be closed. So the water has moved out. The pink image, and again, don't rely on stains. This one is open. Okay, so water's moving in, so it makes it nice and rigid. Here's our equation again. This time we're going to focus on what does it produce. So it's going to produce glucose, and do pause this video to take a look and read all of these little extra side notes. It tells you where it's going to, how it's transported. So just take a moment to take a look at that. So jumping into the light reactions, we're going to look at some molecules. ATP you've heard before. A new molecule you may not have heard of before is NADP. There's other molecules in the process that we're not going to really memorize or focus on in this class, like FAD, FADPH. There's also some coenzymes, and again, you'll tackle that when you get to an upper level class. But some of these we do need to know because it's going to power the next phase or the next set of reactions. One of those is NADP, and that stands for nicotinamide adenine dinucleotide phosphate. And that's a coenzyme, which is kind of like a protein. So on that, let's just focus on NADP. We're going to see an H attached to it. This is going to carry a hydrogen, and that's going to be important to help power this system as we move along. So right now, we're just going to focus on this portion here of the thylakoid membranes. So light is going to come in, light reactions, it requires sunlight. Oxygen is going to be given off, and what else is going to be produced is ATP and this hydrogen carrier molecule, the NADP, and it's going to carry the hydrogen. So I just want to touch on the dark reactions. Here's my equation again. So you can see this time it's written carbon dioxide, water, sunlight, and it's going to make glucose and oxygen. So for the dark reactions, the products from the light reaction is going to help run this particular set of reactions. I've got ATP, NADPH. It's going to react with carbon dioxide, which is right here, and it's going to make glucose. Okay? Light reactions using that water molecule, light, is going to make oxygen. Here's my chloroplast again. This should look very familiar. So chloroplasts inside the plant cells. Here are my thylakoid membranes. A stack of those is called the grana of granum. In between is the stroma. So light reactions in the thylakoid, dark reactions in the stroma. And then, so we've already talked about light and dark reactions, and it takes place in the chloroplast. Okay. Now, photosynthesis does require pigments, and it's activated by the sunlight. So the pigments absorb some wavelengths of light, and it also reflects others depending on the pigment. And so we're going to look at the spectrum, like when you look at a prism, the different wavelengths of light. So chlorophyll has two primary pigments. The main one is red and blue, and it's going to reflect green. So whatever you see, the plant, if I have a green plant, it's reflecting the green pigment or the green wavelength of light. If it appears red, then it's reflecting the red, and those are different names for those different pigments. So there are 5 types of chlorophyll. We have A, B, C, D, and bacterial chlorophyll. I mentioned that in the last lecture. So the two, again, we're going to focus on is A and B. So the difference between the two is chlorophyll A is found in all algae, and chlorophyll B is found in all plants. So that's how you can tell the difference. So A and B are most of our plants in our area. Now, when the plant is not using chlorophyll, it uses something called accessory pigments. It's kind of its backup plan. So what it does is whatever wavelengths of light that chlorophyll cannot absorb, because it can only take in so much, just like a sponge, then it's going to go to these accessory or backup pigments. And then later, it's going to transfer that energy to the chlorophyll molecules, so it'll keep that photosynthesis process running. So part of those accessory pigments, there's a lot of them. I'm just going to touch on a few. So the first one is carotenoid, and that is yellow, orange, browns. Two that we're going to focus on is the orange and yellow color. So orange is carotene, it sounds like carrots, and yellow is xanthophyll. It sounds like a Z, but it's spelled with an X. The other one is phycobilins, and that absorbs the green, violet, blue, and you'll see that mainly in like your blue-green algae. This is this spectrum I was talking about, the wavelengths of light. So chlorophyll it's going to absorb the longer wavelengths, and in this case, it's going to be the red and the blue, okay? And then you'll be able to reflect and see the green color. Digging deeper into light reactions, there are actually Within the thylakoid membrane, there are something called photosystems. And there are two photosystems, photosystem 1 and photosystem 2. And at each location in the membrane, they're absorbing light, they have electrons, they have hydrogen ions, and it's pushing these ions across to make these different molecules. So let's go ahead and look at an illustration. Let's orient ourselves a little bit. So here's my equation again. I'm just going to pop that up there. Here's my chloroplast. So I'm going to take one of these thylakoid membranes. So here's the membrane itself. Okay, this is the inside of the membrane. Surrounding it, this is the stroma. That's that kind of light green from some time ago. So the thylakoid membranes where light reactions take place. The stroma is where dark reactions take place. So these are proteins. This is all proteins actually. So this one protein is called photosystem 2, and then it's going to move to photosystem 1. Well, it seems like it should be photosystem 1 then 2, but it was actually based on the naming of when it was discovered. So system 2 was discovered first and then system 1. So light's going to come in. So these green are the pigments, okay, the light is absorbing the wavelengths of light. The yellow is showing the electrons moving throughout. Here's my water molecule. It's actually going to split to help push these different molecules through. It's called a pump. So when it splits, when a water molecule splits, it's going to release the oxygen into the atmosphere. It's then going to have these hydrogen ions. It's going to help make these other molecules to use for later dark reactions. Then I've got photosystem 1. So its main thing is it's also going to use sunlight. And here is that hydrogen carrier molecule. So NADP is not energized. When I pick up a hydrogen ion and it becomes NADPH, then it's energized. That's going to go, another name for the dark reactions is the Calvin cycle. So don't be thrown that that's a totally different cycle. So this is going to go to the Calvin cycle. Here I've got hydrogen going through this special protein called ATP synthase. Synthase means to make, to synthesize. So it's going to be making ATP, which is also going to go to the Calvin cycle. So the big things you need to know out of the light reactions: where does it take place? What does it need to operate? And what's going to be made? So I've got electrons. When the light hits the chlorophyll, they're going to get excited, energized. So remember I said you can see them shaking under the microscope when it gets warm. Those electrons are going to be passed along the electron transport chain. So that sits in between photosystem 2 and photosystem 1. That water molecule is going to split. It's going to release oxygen to the atmosphere. And then also it's going to make that NADPH to go to the Calvin cycle. But these ions are going to move back and forth across the membrane, and it's going to eventually make some ATP. Here's just another illustration. This time, the ATP synthase, that protein, is sitting at the bottom. The other illustration, it was up at the top. Same thing's going on. Light hitting, splitting a water molecule. This concentration gradient inside is higher, so these hydrogen ions are wanting to move back and forth. This is the electron chain I mentioned. So electrons are pushed along. More light's going to hit this photosystem 1. It's going to make the NADPH, and down here ATP is going to be made. Again, just a different diagram. This one I threw in here. You may want to pause this just to read these illustrations of what's going on in the diagram. I'm not going to repeat that again. And so just one more time, the end products, I've got my hydrogen carrier molecule. I've got my oxygen that's going to be made, my ATP, O2 is released to the environment, NADPH, ATP is going into the chloroplast. More specifically, it's going to go to the stroma, and it's going to be used to power or to start the dark reactions. So again, I know I went through that pretty quickly. This is a short lecture. I wanted to break up light and dark reactions just so you have time to go back and take a look at the light reactions. So knowing where it takes place, knowing the steps, knowing what it needs to operate and what is produced, that is the function of this lecture. So if you have any questions, just let me know and I will see you in class.